Is REM sleep a breathing disturbance? The short answer is yes, but as usual, things are more complicated. I am Tai, and today we talk about sleep, dreams, and breathing patterns. Breathing is irregular during rapid eye movement, REM sleep, and stable during non-REM sleep. Why this is so is not yet known. Breathing, a cyclical phenomenon, is very variable when people are awake. In addition to an intrinsic breath-by-breath variability of resting breathing thought to derive from the complex behavior of the respiratory central pattern generators, breathing is influenced by our activities and emotions, or non-respiratory, speaking or playing music. This is possible by the dual neural control of breathing, with subcortical and cortical networks in constant interplay. Not like wakefulness, breathing becomes more regular while we move into deeper stages of non-rapid eye movement, NREM sleep. In contrast, breathing is erratic in REM sleep. It becomes irregular, faster, and shallower, and even brief central apneas may occur. REM sleep ends a sleep cycle and is characterized by cortical activation, muscle paralysis, atonia, and rapid eye movements. Rich mental imagery, dreams, often accompanies this stage. Why is breathing so disturbed during REM sleep? REM sleep breathing pattern cannot be explained by chemoreceptor or vagal afferent activities because the ventilatory responses to chemical stimuli and other respiratory reflexes are blunted during periods of REM sleep with eye movements. In animals, REM sleep is divided on the basis of behavioral characteristics into tonic REM sleep, during which the animal sleeps peacefully, and phasic REM sleep, when the animal exhibits twitching movements of the ears, muzzle, and limbs with frequent eye movements. In dogs, the ventilatory response to CO2 is diminished during phasic REM sleep, in comparison with that during either tonic REM or slow-wave sleep. In cats, eye movements and pontogeniculo-occipital waves are associated with alterations in activity of medullary respiratory neurons. These observations suggest that the subdivision of REM sleep may be linked to breathing. In humans, there are no clear behavioral characteristics apparent during REM sleep. A sharp distinction based on eye movement frequency is usually not possible, as most subjects exhibit a wide spread of eye movement densities. However, there is some indirect evidence from studies in humans suggesting that eye movements might be associated with breathing disturbance. Studies have shown that arterial desaturation in patients with chronic obstructive pulmonary disease was more severe and frequent during periods of REM sleep, associated with eye movements, than during periods without eye movements. In cats, the activity of medullary respiratory neurons increases in REM sleep compared to NREM sleep, suggesting the existence of an excitatory drive to the respiratory central pattern generators in REM sleep. This excitatory drive persists when spontaneous breathing is eliminated by mechanical ventilation, but its neural origin is still unclear. Pontine-dependent processes could cause the co-activation of the multiple phasic events that occur during REM sleep. In support of this idea, discharge rates of medullary neurons, whose activity is highly specific to REM sleep, correlate with eye movement bursts and erratic breathing. The activity of medullary respiratory neurons in REM sleep is related to the density of pontogeniculo-occipital waves, a hallmark of REM sleep. However, pontine cats, in which the structures above the pons have been removed, have regular and not irregular breathing in REM sleep. This suggests that suprapontine structures cause the breathing irregularities observed in normal cats in REM sleep. The nature of these structures is not known. Many suprapontine structures modulate the respiratory neural outflow generated by the central breathing pattern generators during wakefulness. These structures include the caudal hypothalamus, the periaqueductal gray area, and midbrain regions, all of which are involved in the coupling of respiration with locomotion. Limbic and paralimbic networks comprising the amygdala, the anterior and dorsal cingular cortices, and the insular cortex are involved in emotional breathing modulation. Also, cortico-subcortical networks can influence breathing control through bulbar projections or through direct corticospinal projections bypassing the central pattern generators. In this way, breathing changes can be observed in response to mental imagery during wakefulness, where, for example, imagining a physical movement such as running is enough to increase ventilation. In accordance with ventilation changes observed during imagined exercise, researchers hypothesized that REM sleep-related breathing irregularities depend on cortical projections to medullary and/or spinal respiratory motor neurons and are correlated with the mental imagery of REM sleep dreams. Whether the phasic properties of REM sleep, such as eye movements or respiratory events, are related to dream imagery rather than random markers of brainstem activation is debated since the discovery of REM sleep. One study found large respiration amplitude, but not respiration variability, correlated with intensive active participation in the dream. In another study, participants' likelihood to report a dream after REM sleep was positively correlated with rate and variability of breathing immediately preceding the awakening. This relationship was stronger if the mental content was judged as vivid, emotional, and involved physical activity. These correlational studies have significant methodological biases because of their retrospective nature, including forgetting, reconstruction of missing fragments, censorship, and the impossibility of temporally coupling a dream report with its corresponding segment in the sleep recording. In addition, subjects have usually had no control over the dream scenario. To overcome these difficulties, a 2018 study used a unique model, subjects that suffered from narcolepsy and were lucid dreamers. Unlike common dreamers, sleepers of lucid dreams are aware that they are dreaming and can exert control over the dream scenario. Because eye movements are spared the muscle paralysis characteristic of REM sleep, lucid dreamers can communicate with an ocular code. This enables the isolation of beginning and the end of a dream episode on the physiological sleep recording. 21 patients with narcolepsy, who had the ability to lucid dream in REM sleep, participated in the study. They were asked to influence their dream content when they reached lucidity. Depending on the nap, they had to imagine a scenario involving an apnoea, diving underwater, vocalizing or yodeling in their dreams. These types of breathing behaviors involve descending inhibitory cortical inputs to the medullary respiratory central pattern generators to produce breath holding, or to avoid the unwanted interruption of a vocal utterance by automatic inspiration. They can also involve excitatory corticospinal inputs to produce preparatory breaths. Sleep and respiration were monitored during multiple naps. Most participants, 86%, were able to signal lucidity in at least one nap. In 50% of the lucid naps, clear congruence was found between the dream report and the observed breathing behavior and, in some cases, a preparatory breath before the respiratory behavior. These results suggest that the cortico-subcortical networks involved in voluntary respiratory movements are preserved during REM sleep and reflect dream content. So, yes, your sleeping breathing pattern is disturbed so it can match your dream, and while doing so, it enables you to move as you like, without falling out of bed. As always, I leave you with blessing for a good breathing day and night.